Hello, everyone. Welcome to our new topic about Mendelian genetics and genetic mapping. We are going to talk about just extensions of Mendelian genetics and also chromosome mapping in eukaryotes. Uh, Tenets for the transmission of heritable characteristics from parents to offspring was developed by Gregor Mendel, and I am sure you are familiar with these photos because you have seen this in many textbooks during your school studying. Now let's talk about just two laws of the Mendelian genetics. These are law of segregation, and this is law of independent assortment. A phenotypic variation observed for various traits is the result of alternative versions of genes at specific genetic loci, which are known as alleles. The offspring inherit one allele at each locus from each of the parents. And you also need to know what are the terms like dominant allele and recessive allele. Dominant alleles, which are denoted by uppercase letter as in, for example, capital A, are those that when co-inherited with another allele dictate the phenotype of the offspring. Whereas the recessive alleles are those that when co-inherited with another allele have no tangible effect on the phenotype. And recessive alleles are denoted by a lowercase letter, for example, this lowercase a in here. The offspring may inherit the same allele from each of the parents, like both of them can be uppercase letter like here or lowercase letter like this one. And in this case, they are called homozygous, or when they get the different alleles from each parent, for example, this combination or this one, in this case, they are called heterozygous. The two alleles for each gene segregate during gamete production. This is law of segregation, when two alleles for each gene segregate during gamete production. Law of independent assortment is another law of Mendelian genetics, so what is important in it? The inheritance pattern of one trait is independent and it does not influence the inheritance of another trait. The independent assortment occurs during anaphase 1 of meiosis after crossover has occurred, and genes may be linked together because of their physical proximity on the chromosomes and therefore may segregate together to affect phenotype. So what does this mean? For example, when the genes are located close to each other, they are very likely to be inherited together. What is the Punnett square? This is a diagram predicting the outcome of a mating between individuals with different genotypes. It provides a graphic representation of all possible genotypes of the offspring of the cross. The interpretation of such data leads to the determination of the frequency of offspring affected by various modes of transmission. And the simplest example is the mating of two individuals who are heterozygous at a single genetic locus. By convention, the two maternal alleles are written over the columns. So let's do this table here. 1, 2. So by the convention, two maternal alleles are written over the columns and paternal alleles are written in the side rows. The genotypes and the phenotypes of the offspring are determined by the combination of the maternal and paternal alleles. In the mating of heterozygotes, the probability of an individual offspring having the genotype AA is 25%, heterozygous like uppercase and lowercase a is 50%, and small a is 25%. And I will show you this right now, how this happens. This is just a simple table here. These are just the parents here, and this is the offspring I am just writing now. This is AA, capital A, lowercase a. Also capital A, lowercase a, and small a, small a. Now let's see the percentage in this offspring. So as you see, uppercase A double is just only 1/4, so it is 25%, which is written in here. This version is found in just half of the offspring. So it is 50%, as you see. And this one is just one in this four offspring, so this is 25%. 75% of the offspring will have the dominant phenotype because it just contains this uppercase A, as you see. So every 3 from the 4, so this is 75%, and only 25% will have the recessive phenotype. Yes, because you know that the recessive phenotype is only expressed when it is the combination like this, homozygous combination. So this was information about Punnett square. Now let's talk about genetic mapping. So genetic mapping, what does this mean? This is just finding the location of each gene on the chromosome. Yes, just how can we do this? Before talking about this, I am going to start with genetic recombination. What is it? You know that during meiosis, chromosomes exchange segments. The child inherits a patchwork of paternal chromosomes. So this is patchwork. Yes, it contains the segments from this one, and also the same happens in here. And this is because of just exchanging the segments in meiosis during conjugation and crossing over in prophase 1 of the meiosis 1. These chromosomes are never a copy of parental chromosomes. What is independent assortment? Let's suppose that father have 2 alleles, F and M genes, in just different in the forms like capital F and lowercase f, and also the same in case of M. F and M are found on different chromosomes. In this case, there will be independent assortment because they are located in different chromosomes far from each other. So occurs if F and M gametes can independently recombine, and there will be 25% chance of each combination in gamete, as you see on this photo. So because these F and M are located on different chromosomes, they can give us these 4 combination. Yes. Now let's talk about if these F and M are located on the same chromosome. So what if the genes are located on the same chromosome? Like both of them are located on chromosome 1, for example, F in the proximal segment of the chromosome 1, M in the distal segment of the chromosome 1. If they are very far apart from each other, the crossover may occur in meiosis. And if they are very far apart from each other and crossover will occur, the result will be same combinations as it was in the separate chromosomes. As you see, we will have these 4 combinations. But what if the genes are located on the same chromosome and they are too close to each other, so that if crossing over occurs, the chances very little, but if it occurs, they will just go together on another chromosome. So in this case, there is low occurrence of recombination. So if the alleles are very close to each other, there is very little chance of crossover and getting the recombination. So we will have the same combinations as was in the parental version. So let's do like a summary in here. So these are the chromosomes here. As you see, some genes are located close together, like this one, 2 neighboring, and some, for example, A and C or B and C are located far apart. Yes, so it is very likely to be a mutation, to be a splitting in this bigger area then this smaller, yes. So if any break occurs here, then A and B will recombine, but there is very little chance that this will happen. If any break here happens, it allows B and C to recombine, and the chance is much higher because they are far away to each other. Now let's talk about recombination frequency. This is frequency of recombined genes, like for example, FM and FM on this photo, yes. So recombination frequency, this will be, in this case, 50% because half of the products have the same combination as it was in the parent, and half have different because of the combination. So recombination frequency is frequency of recombined genes, and it is denoted by Greek letter theta, and it ranges from 0 to 0.5. The key point here is that recombination frequency is proportional to distance, which means that the closer the genes locate to each other, the recombination frequency is smaller. But the bigger the recombination frequency, the distance is bigger or longer between the genes. This is used for genetic mapping to identify the location of the genes according or toward each other. So when the genes are located close together, the recombination frequency is close to 0. Yes, when they are close together, the smaller the distance between them. When they are far apart each other, the recombination frequency is higher, and it can be as maximum as 0.5. And this is used for genetic mapping of genes. We can calculate the recombination frequency, and then we can, by this, identify the distance between the genes from this recombination frequency. Now let's talk about what is linkage mapping. This is done by studying families, and we track frequency on genetic recombination and use the frequency to determine relative gene location. For example, we can study the genes A, B, and C, and we can just calculate the frequency of their combinations. After we have these frequencies, we know that recombination frequency is proportional to distance, and from this frequency we can identify how they can be located on the chromosome. Like you see that 0.16 is frequency of combination A and B, and 0.08 and 0.08 is frequency between A, C, and CB combinations. Yes, so this is much bigger, which means that A and B are located farther from each other than A and C or C or B. Yes, so A and B far each other, and the same distance between C and A and C and B. Now let's talk about what is linkage. Linkage is tendency of alleles to transmit together. When there is more linkage, there is less independent assortment. When they are close together, the theta is 0, and this is also equal that they are tightly linked. When they are far apart, the theta can be as high as maximum like 0.5, and this means they are unlinked. Now let's talk about linkage disequilibrium. For this, we use to study Now let's talk about what is linkage disequilibrium. This is used to study genes that are very close together, and recombination is very rare. And in this case, family studies are impractical, so this is done by studying large populations. Before talking about linkage disequilibrium, let's talk what is linkage equilibrium. Let's suppose that gene A has two polymorphisms, this one and this one. And let's also suppose that A is found in 50% of individuals and small a is found in 50% of individuals. In this case, the frequencies of A and small a will be 0.5 and 0.5, yes. And gene B, let's suppose, has two polymorphisms like uppercase B and lowercase b. let's suppose that B uppercase is found in 90% of individuals, and smaller case B will be in 10% of individuals. So their frequencies will be for capital B, it will be 0.9, and for small b, it will be 0.1. The linkage disequilibrium, so we can from this calculate the frequencies of the combinations this A and B can give us, like just frequency of combination of A, B will be 0.45. For this combination, the frequency will be also 0.45, and for this combinations, the frequency will be 0.05. And this is just linkage equilibrium if this calculation is the same as it is found on the population we are studying. But in linkage disequilibrium, the population frequencies are higher or lower than expected. I mean, when we have calculated this one, and this is different than the real combinations, the frequencies in the population, this is called linkage disequilibrium. For example, AB, if it is found in real population 0.75 or which means higher than expected, which we calculated 0.45, this is linkage disequilibrium. And this haplotype AB is a linkage disequilibrium. During linkage disequilibrium, we need to consider new gene mutation. So why? Because during new mutations, there is a bigger, or the chance of the splitting between the genes is higher. So it increases the chance of recombination. That's why we usually need to assume new mutation for linkage disequilibrium. And please memorize that linkage disequilibrium is affected by genetic distance and the time the alleles have been present in population. Different populations have different degrees of linkage disequilibrium. This concludes our topic about genetic mapping and Mendelian genetics.